Hi everyone. Today our topic is bioequivalence study design and IVIVC. IVIVC means in vitro in vivo correlation. The bioequivalence definition is it refers to the drug substance in two or more identical dosage forms reaches systemic circulation at the same rate and to the same relative extent. That is, their plasma concentration time profiles will be identical without significant statistical differences. Some introduction about this bioequivalence is during last 4 decades there is an increased use of generic drug products in order to lower the healthcare cost. With increased bioavailability and use of generic drug products, healthcare professionals are encountered with a large number of multi-source products from which they have to select therapeutically equivalent products. Generic substitution is of concern not only for the healthcare professionals but also for pharmaceutical industries. Consumers and government officials may research papers have pointed out the concern regarding standards for approval of generic products which may not always ensure therapeutic equivalence. Generally, demonstration of bioequivalence (BE) is the most appropriate method of ensuring therapeutic equivalence between two medicinal products. Bioequivalence studies should be conducted for comparison of medicinal products containing same active substance. Such studies need to be carefully designed to take into account biopharmaceutical and ethical, medical, pharmacokinetic, and analytical statistical consideration. The study should be aimed to critical assesses the possibility of alternate use the products. As per the FDA guidelines, bioequivalence is defined as the absence of significant difference in the rate and extent to which active ingredient or active moiety in pharmaceutical equivalents or pharmaceutical alternatives becomes available at the site of drug action when administered at the same molar dose under similar conditions in inappropriately designed studies. One more definition is the rate and extent to which the active ingredient or active moiety is absorbed from a drug product and becomes available at the site of action. For drug products that are not intended to be absorbed into the bloodstream, bioavailability may be assessed by measurements intended to reflect the rate and extent to which active ingredient or active moiety becomes available at the site of action. Bioequivalence and bioavailability studies are important during drug development of both new drug products and their generic equivalents. Provision of bioavailability or bioequivalence study data is an important element in support of investigational new drug applications is called INDs and NDAs. NDAs means new drug applications. Abbreviated new drug applications means ANDAS. Here we have three types. One is INDS. INDS means new drug applications. NDAS means new drug applications. And ANDA is abbreviated new drug applications and their supplements. The term generic drug product has been defined as interchangeable multi-source pharmaceutical product. Again I repeated, the term of generic drug product has been defined as interchangeable multi-source pharmaceutical product. Generic products are the copies of branded name drugs with the same dosage form, strength, route of administration, intended use, and toxicity profile as the original innovator drug. Concern about lowering healthcare costs has resulted in an increase in the use of cheaper generic drug products instead of branded products. The innovator drugs are protected from copying by patents that last for 20 years from the first filling of the new chemical entity. Many people are concerned why generic drugs are often cheaper than the branded name versions. It is because all the development work and clinical trials on new chemical entity are carried out by innovator to get initial drug approval, which is later on reflected in its high prices, whereas the generic manufacturer only need to submit the bioequivalence data of the generic product to get a product license. The new products need to undergo bioequivalence testing before they are marketed. The difference may exist in absorption reflected in differing bioavailability profile of various brands. Production batches are dosage forms of a drug. This can lead to either over or under medication of one entity in substituted for the other. The under medication can lead the therapeutic failure, and on the other hand, over medication can lead to toxicity. To avoid such risk, it is best to study the bioavailability of all products, but particularly it is not possible. So each drug and any change in formulation must be considered individually while keeping in mind the real medical need for such studies in order to ensure efficiency and safety of these drugs. Many clinicians, while switching or interchanging the different products, are concerned with the safety and effectiveness of the new drug. This concern is because of the fact that small changes in bioavailability can lead to significant changes in the efficacy or safety of drug. Bioequivalence studies are designed with this concern in mind and to devise the strategies that minimize the risk to the patient. So when the generic product is pharmaceutically equivalent as well as bioequivalent to the innovator drug, then it is expected to the therapeutically equivalent. First of all, we should know what is the difference between branded drugs and generic drugs. So here I will show one video, you can understood easily. Please follow this video. We have three types of equivalence. So first one is pharmaceutical equivalence, second one is therapeutic equivalence, and third one is clinical equivalence. Pharmaceutical equivalence definition is drug products are considered to be pharmaceutical equivalents if they contain the same active ingredients and are identical in strength or concentration, dosage form, and route of administration is called pharmaceutical equivalence. Next, therapeutic equivalence is defined as it indicates that two or more drug products that contain the same therapeutically active ingredient and elicit identical pharmacological effects and can control the disease to the same extent. Again, I repeated, it indicates that two or more drug products that contain the same therapeutically active ingredient elicit identical pharmacological effects and can control the disease to the same extent. Next, clinical equivalence defined as when the same drug from two or more dosage forms gives identical in vivo effects as measured by a pharmacological response or by control of a symptom or a disease is called clinical equivalence. Here we have one question: when we do our bioequivalence studies, first one is clinical service form to final market form. Second one is change of formulations, means capsules to tablet. Third one is generic formulations. Fourth one is change of process or manufacturing site. Fifth one is regulatory requirement. And the sixth one is establishment of pharmacokinetic parameters. And the seventh one is study of formulations and process variables. In that time only we can do this bioequivalence studies. We can also face some problems in bioequivalence, that are active drug ingredient has low solubility in water, dissolution rate is low, certain structural forms of active drug ingredient, a drug product that have high ratio of excipients to active ingredients, specific ingredients such as hydrophilic and hydrophobic excipient and lubricant may interfere with absorption. Active ingredients observed in particular segment of GAT, rapid metabolism in intestinal wall or in liver, drug absorption process. We have some limitations in bioavailability and bioequivalence studies, that are difficult for drugs with a long elimination half-life. Highly variable drugs may require a far greater number of subjects. Drugs that are administered by routes other than the oral route. Drugs are dosage forms that are intended for local effects have minimal systemic bioavailability. Examples ophthalmic and dermal, etc. Biotransformation of drugs make it difficult to evaluate the bioequivalence of such drugs. Examples stereoisomerism. Advantages of bioequivalence: to minimize the effect of intersubject variability, and it minimizes the carry-over effect and requires less number of subjects to get meaningful results. Disadvantages for bioequivalence: requires longer time to complete the studies, and completion of studies depends on number of formulations evaluated in the studies. Increase in study period leads to high subject dropouts, and medical ethics does not allow to many trials on a subject continuously for a longer time. Before we are going to discuss another topics, first of all we should know understanding the clinical trials. Okay, so please go through this video, you can easily understand.
Clinical trials are research studies that evaluate new ways to improve treatments and quality of life for people with diseases like pancreatic cancer. Clinical trials are an option for all patients to consider and play an important role in improving healthcare. Results from clinical trials help develop new drugs, new diagnostic tools, and new clinical procedures. Clinical trials can also spur new questions for research, which lead to new discoveries over time and help us to better understand diseases like pancreatic cancer. All the drugs and tools we have today were made possible because patients volunteered for clinical trials. Every clinical trial has what are called eligibility criteria. For example, a patient may need to have a certain medical condition or gender or other specifics to meet the criteria of participating in the clinical trial. Otherwise, they are not eligible to join that clinical trial and could seek another trial that better fits their situation. Patients who want to volunteer need to understand the potential benefits and risks of participation in a clinical trial. Patients may personally benefit from receiving new therapies or procedures. They receive close supervision from clinical staff. Sometimes the cost of the new drugs or testing is free or waived. Participants may also be part of a research breakthrough that helps thousands of future patients. Patients face the risk that they may not benefit from the new treatment. Additionally, participating in a clinical trial requires commitment. Trials can be lengthy or demanding and may require extra treatments, extra time in the clinic, or tests or costs that are not part of standard care. It is possible, if the study is randomized, that they may be part of the control group that does not receive the new drug being studied. Informed consent is a process that helps patients make an informed decision about whether to join a clinical trial. An informed consent document will be provided that explains the purpose, procedures, and potential benefits and risks of the trial. There will also be thorough discussions between the doctor and patient so that all questions can be clearly answered. In late-stage clinical trials, participants are generally split into two groups at the beginning of the study. One group receives the standard treatment plus the new treatment. This group is called the treatment group. The other group receives the standard treatment only and does not receive the new treatment. This group is known as the control group. A computer is used to randomly assign the patient to either the treatment group or the control group. Randomization helps to prevent bias because neither the patient nor the doctor can choose which treatment group a patient is assigned to. Some clinical trials may want to study the effectiveness of adding a new treatment to a standard treatment to see what works better. In these studies, half the patients will receive the standard treatment plus the new treatment, and the other half will receive the standard treatment plus a placebo. A placebo is a treatment that looks identical to the new treatment, but contains no active ingredient. These types of studies are often called blinded, meaning that one or more parties involved in the trial do not know which patients have been assigned to which treatment. Single-blinded means that only one party, either the researchers or the patients, knows which patients have been assigned to which treatment. Double-blinded means that both the researchers and the patients do not know which patients have been assigned to which treatment. The strongest, most reliable results come from clinical trials in which the treatment is double-blinded, the patients are randomized, and the study has a control group. This is called a double-blinded, randomized, controlled clinical trial. Clinical trials are carefully monitored to protect patients, and several layers of review committees are involved to protect patients from any possible harm. Patients can leave a clinical trial at any stage. However, they should discuss this with their doctor before exiting to ensure the safest procedures are followed. There are 4 different phases of clinical trials. Safety is emphasized in each phase. If a trial does not have a positive or safe outcome in Phase 1, then it will not be allowed to proceed to a Phase 2 trial, and so on. Before a Phase 1 trial begins, there is a Phase 0, where the new treatment has already been found to be safe in animals or healthy human volunteers. In Phase 1 clinical trials, a new treatment is tested in a small group of participants for the first time to determine a safe dose and see how the treatment affects the body. Phase 2 is conducted with a larger patient group than the previous phase and studies how well a new treatment or combination of treatments works for a certain type of cancer, like pancreatic cancer. At this stage, the eligibility criteria become more restrictive. Phase 2 studies can be either randomized or non-randomized. Phase 3 trials are conducted in an even larger group of participants. This phase involves a randomized design, comparing the new treatment to the standard treatment. If a Phase 3 clinical trial is positive, typically the new treatment will be approved and licensed for use in the United States by the Food and Drug Administration, or FDA. After the treatment has been approved, Phase 4 clinical trials involve monitoring to continue to assess safety, effectiveness, and optimal use. Clinical trials play an important role in moving science forward and can be a hopeful option for many patients with pancreatic cancer. To learn more about clinical trials and
We have two types of bioequivalence studies. One is in vivo and second one is in vitro. In this in vivo bioequivalence studies are the following sequence of criteria is useful in assessing the need for in vivo studies. First one is oral immediate release products with systemic action. Second one is non-oral immediate release products. Third one is modified release products with systemic action. If it comes first one, oral immediate release products with systemic action, under this indicated for serious conditions requiring assured response, narrow therapeutic margin, and pharmacokinetics complicated by absorption and unfavorable physicochemical properties, examples low solubility, metastable modification, instability, etc. Documented evidence for bioavailability problems will be occur. No relevant data available unless justification by applicant that in vivo study is not necessary. In vivo bioequivalence studies are conducted in the usual manner as discussed for bioavailability studies, that are the pharmacokinetic and the pharmacodynamic methods. First one, pharmacokinetic methods. In this pharmacokinetic methods, we studied plasma level time studies, and second one is urinary excretion studies. And pharmacodynamic methods are involved in this acute pharmacological response, and second one is therapeutic response. These are the mainly in vivo bioequivalence studies. Next one is in vitro bioequivalence studies. In this, if none of the above criteria is applicable, comparative in vitro dissolution studies will sufficient. In vitro studies, that is dissolution studies, can be used in vivo bioequivalence under certain circumstances called as bio-waivers, means exemptions, that are the drug product differs only in strength of the active substance it contains, provided all the following conditions hold, like pharmacokinetics are linear and the qualitative composition is the same, the ratio between active substance and the excipients is the same, or in the case of small strengths, the ratio between the excipients is the same. Next, both products are produced by the same manufacturer at the same production site. Next one, a availability or bioequivalence study has been performed with a original product. Next, under the same test conditions, the in vitro dissolution rate is the same. Next, the drug product has been slightly reformulated, or the manufacturing method has been slightly modified by the original manufacturer in ways that can convincingly be argued to be irrelevant for the bioavailability. The drug products meets all the following requirements, that are the product is in the form of solution or solubilized form, like elixirs, syrup, tinctures, etc. The products contain active ingredient in the same concentration as the approved drug product. The product contains no excipients known to significantly affect absorption of the active ingredient. Next, main important thing is an acceptable IVIVC. IVIVC means in vitro in vivo correlation, and the in vitro dissolution rate of the new product is equivalent with that of the already approved medicinal product. Moreover, the product is intended for topical administrations like creams, ointment, gels, etc. for local effect. The product is for oral administration but not intended to be absorbed like antacid or radio-opaque medium. The product is administered by inhalation as a gas or vapor. The criteria for drug products listed above indicate that bioavailability and bioequivalence are self-evident. Next topic is IVIVC. IVIVC is nothing but in vitro in vivo correlation. Definition of IVIVC. Dissolution is the rate limiting step for absorption of controlled release or extended release formulations. The establishment of a relationship between the release of the drug in vitro and its release in vivo or its absorption into the systemic circulation is IVIVC. IVIVC అంటే మనం మెయిన్గా రెండింటి మధ్య కోరిలేట్ చేస్తున్నాం. ఒకటి వచ్చేసి డిజల్యూషన్, ఇంకొకటి వచ్చేసి అబ్జార్ప్షన్ ఇంటూ ద సిస్టమిక్ సర్క్యులేషన్. ఫర్ ఎగ్జాంపుల్, మనం ఏదైనా ఎక్స్టెండెడ్ రిలీజ్ టాబ్లెట్ కానివ్వండి, లేకపోతే కంట్రోల్డ్ రిలీజ్ టాబ్లెట్ కానివ్వండి, దీన్ని డిజల్యూషన్ రేట్ తెలిస్తే మనకి ఈజీగా సిస్టమిక్ సర్క్యులేషన్లో అబ్జార్ప్షన్ ఎంత అనేది మనకి ఈజీగా తెలుస్తుంది. సో దీన్ని రెండింటి మధ్య డిఫరెన్స్ ఏందని తెలుసుకోవడాన్ని మనం ఐవిఐవిసిని యూజ్ చేస్తాం అన్నమాట. ఇక్కడ ఇన్ విట్రో ఇన్ వివో అంటే ఇన్ విట్రో మీన్స్ మనం డిజల్యూషన్ రేట్ని చెక్ చేసే ఆపరేటర్స్ని ఏదైతే యూజ్ చేస్తున్నామో దాన్ని ఇన్ విట్రో అంటాము. ఇన్ వివో అంటే మన బాడీలో సిస్టమిక్ సర్క్యులేషన్లో ఎంత అబ్జర్వ్ అవుతుంది చెక్ చేయడాన్ని మనం ఇన్ వివో అంటాం. నెక్స్ట్, ద మెయిన్ ఆబ్జెక్టివ్స్ ఆర్ సిగ్నిఫికెన్స్ ఆఫ్ IVIVC. The main objective of developing and evaluating an IVIVC is to enable the dissolution test to serve as a surrogate. It reduces the number of bioequivalence required for approval as well as during scale up and post approval changes, that is called SUPAC. IVIVC shortens the drug development period, economizes the resources and leads to improved product quality, and a means of assuring the bioavailability of active ingredients from a dosage form, supports and or validates the use of dissolution methods and specifications. We have two main basic approaches. First one is by establishing a relationship between the in vitro dissolution and the in vivo bioavailability parameters. Second approach is by using the data from previous bioavailability studies to modify the dissolution methodology in order to arrive at meaningful in vitro in vivo correlation. These are the parameters for correlation involved in vitro and in vivo. We have different levels of correlation in IVIVC. In that, we have different types of levels. First one is level A, level B, level C, and multiple C. If we check in level A correlation, highest category of correlation, linear correlation, superimposable in vitro and in vivo input curve are can be made superimposable by use of a constant offset value. So most informative and useful from a regulatory perspective. Next, level B correlation is uses the principles of statistical movement analysis. Second one is the mean in vitro dissolution time is compared either to the mean residence time, that is called MRT. MRT means mean residence time, or to the mean in vivo dissolution time. Is not a point to point correlation. and level B correlations are rarely seen in NDAS. Already we discussed about NDAS. Next, level C correlation represents a single point correlation. One dissolution time point is compared to one mean pharmacokinetic parameter such as AUC, Tmax or Cmax. So weakest level of correlation as partial relationship between absorption and dissolution is established. Last one, multiple level C correlation is multiple level C correlation relates one or several pharmacokinetic parameters of interest, that is Cmax, AUC or any other suitable parameters, to the amount of drug dissolved at several time points of the dissolution profile. Its correlation is more meaningful than that of level C as several time points are considered. I hope you understood Unit 3 bioavailability and bioequivalence. If you have any doubts, please comment in comment section. I will clarify you. Thank you so much.